Today we are going to discuss about digestive system part 2. In this part we are going to discuss about the movements of GIT, physiology of digestion and absorption, acid production in the stomach, digestion and absorption of nutrients, disorders of GIT. At first, let's discuss about the movements of gastrointestinal tract. First one, mastication. Mastication or chewing is the first mechanical process in the gastrointestinal tract by which the food substances are torn or cut into small particles and crushed or ground into a soft bolus. Significances of mastication. First one, breakdown of foodstuffs into smaller particles, mixing of saliva with food substances thoroughly, lubrication and moistening of dry food by saliva so that the bolus can be easily swallowed. Fourth one, appreciation of the taste of the food. Control of mastication. Action of mastication is mostly reflex process. It is carried out voluntarily also. The center for mastication is situated in medulla and cerebral cortex. Muscles of mastication are supplied by the mandibular division of fifth cranial trigeminal nerve. Deglution. Deglution or swallowing is the process by which food moves from the mouth into the stomach. Stages of deglution. It occurs in 3 stages. First one is oral stage, second one is pharyngeal stage, oral stage, pharyngeal stage, esophageal stage. First one, oral stage, when food moves from the mouth to the pharynx. Second one, pharyngeal stage, when the food moves from the pharynx to the esophagus. Esophageal stage, when the food moves from the esophagus to the stomach. And next, movements of stomach. Activities of smooth muscles of stomach increase during gastric digestion when the stomach is filled with the food and when the stomach is empty. Types of movements in stomach. First one, hunger contractions. Second one, receptive relaxation. Third one, peristalsis. Filling and emptying up of stomach. Filling of stomach. While taking food, it arranges itself in the stomach in different layers. The first eaten food is placed against the greater curvature in the fundus and the body of stomach. The successive layers of food particles lie nearer the lesser curvature until the last portion of the food is eaten. Emptying up of stomach. Gastric emptying is a process by which the chyme from stomach is emptied into the intestine. Food that is swallowed enters to the stomach and remains there for 3 hours. During this period, digestion takes place. Movements of small intestine. They are essential for the mixing and the chyme with digestive juices, propulsion of food and absorption. Types of movements of small intestine. There are of 4 types. Mixing movements, segmentation movements, pendicular movements, propulsive movements, peristaltic movements, peristaltic rush, peristalsis in fasting, migrating in motor complex, movements of villi. Movements of large intestine. Usually the large intestine shows the sluggish movements. Still these movements are important for mixing, propulsive and absorptive functions. Types of movements of large intestine. There are of 2 types. Mixing movements involves segmentation contracts. Second one, propulsive movements, mass peristalsis. Defecation. Voiding feces is known as defecation. Feces is formed in the large intestine stored in the sigmoid colon. By the influence of appropriate stimulus, it is expelled out through the anus. This is prevented by tonic constriction of anal sphincters in the absence of the stimulus. As discussed in the below diagram, it is shown in the diagram A, the contraction of the bolus. Next, B involves the stimulus tonic constriction. Next, the anal sphincter. C, absence of the stimulus. Next, we are going to discuss about physiology of digestion and absorption. Digestion of food in alimentary canal. Mouth. The breaking down of food takes place in mouth by chewing or mastication. Teeth and muscle helps in mastication. Salivary amylase from parotid and submandibular glands begins carbohydrate digestion. It breaks the complex sugars like polysaccharides into disaccharides such as maltose, and isomaltose. Digestion in stomach. Stomach cells produce gastric secretions which helps to continue chemical digestion in the stomach. Pepsinogen is secreted by the chief cells that combines with the hydrochloric acid secreted by the perotyl cells into to form pepsin. Pepsin along with the hydrochloric acid breaks proteins into smaller polypeptides which travel into the small intestine. The digestion in small intestine is carried out by the pancreatic enzymes and the intestinal enzymes. Let's see the pancreatic enzymes. Pancreatic enzymes and functions, and the given product is given. First, pancreatic enzyme trypsin or chymotrypsin act upon the proteins. It forms smaller peptides. Pancreatic lipase breakdown of triglycerides into free fatty acids and monoglycerides. Pancreatic amylase breaks down of carbohydrates into the maltose and oligosaccharides. Carboxypeptidase breaks down into the proteins. Carboxypeptidase helps in breakdown of proteins, forms the peptides and amino acids. Nucleases breaks nucleic of acids, produces nucleases. Maltose acts on maltose, produces glucose. Sucrose acts on sucrose, produces glucose and fructose. Lactase acts on lactose, produces glucose and fructose. Aminopeptidases and dipeptidases breaks the peptide chain, produces peptides and amino acids. Nucleosidase and phosphofatase breaks into to form peptones, phosphates and nitrogens. Absorption of food in the alimentary canal. Absorption in the mouth. Absorption of food does not take place in mouth. However, some chemicals like nitroglycerin and some vitamins can be absorbed. Absorption in stomach. There is little absorption in the stomach as the food is not digested completely. Water, alcohol and lipid soluble drugs are absorbed in the stomach. Absorption in the small intestine involves glucose and galactose are absorbed through the intestinal villi cells. Monosaccharides are absorbed by the facilitated diffusion. Amino acids and dipeptides are also absorbed through the villi cells by the facilitated diffusion. Glycerol and fatty acids are absorbed through the villi cells. Vitamins are absorbed by the small intestine via simple diffusion. Mineral absorption takes place throughout the entire small intestine. Absorption in large intestine. The absorption is complete when the material reaches the large intestine. Water and the certain salts are absorbed through the mucous membranes of large intestine. Acid production in the stomach. Hydrochloric acid secretion is an active process that takes place in the canculi of the perotyl cells in the gastric glands. Carbon dioxide is derived from the metabolic activities of perotyl cells. It combines with water to form carbonic acid in the presence of carbonic anhydrase. This enzyme is present in high concentration in perotyl cells. Carbonic acid is the most unstable compound and immediately splits into hydrogen ion and bicarbonate ion. The hydrogen ion is actively pumped into the cannulas of perotyl cells. Simultaneously, the chloride ion is also pumped into the cannulas of actively. Nerve supply of GIT. Extrinsic nerves that control enteric nerve system are from the autonomous nervous system. They are sympathetic nerve fiber. Preganglionic sympathetic nerve fibers to the GI tract arise from the lateral horns of the spinal cord between the fifth thoracic and the second lumbar segment T5 to L2. Here the fibers leave the spinal cord, pass through the ganglia of sympathetic chain without having any synapse and then terminate in the celiac mesenteric ganglia. The postganglionic fibers from these ganglia are distributed throughout the GI tract. Digestion and absorption of nutrients like carbohydrates, proteins and lipids. Digestion of carbohydrates in the mouth. Enzymes involved in the digestion of carbohydrates are known as amylolytic enzymes, the only amylotic enzymes present in the saliva. The salivary amylase or ptyalin in the stomach. Gastric juice contains a weak amylase which plays a minor role in digestion of carbohydrates. In the intestine, amylolytic enzymes present in the small intestine are derived from the pancreatic juice and succus entericus. Amylolytic enzyme in pancreatic juice. Pancreatic juice contains pancreatic amylase. Amylolytic enzymes in succinic entericus. Amylolytic enzymes present in succinic entericus are maltase, sucrase, lactase, dextrinase and trehalase. Absorption of carbohydrates. Carbohydrates are absorbed from the small intestine mainly as monosaccharide via glucose, galactose and fructose. Absorption of glucose. Glucose is transported from the lumen of small intestine into the epithelial cells of the mucous membrane of small intestine by means of sodium co-transport from the epithelial cells. Glucose is absorbed into the portal vein by the facilitated diffusion. However, the sodium ion moves laterally into the intercellular space. From here, it is transported into the blood by active transport utilizing the energy liberated by the breakdown of adenosine triphosphate (ATP). Absorption of galactose. Galactose is absorbed from the small intestine in the same mechanism of that glucose. Absorption of fructose. Fructose is absorbed into the blood by means of facilitated diffusion. Some molecules of fructose are converted into glucose. Glucose is absorbed described above. Digestion and absorption of proteins. Digestion of proteins. Enzymes responsible for the digestion of proteins called proteolytic enzyme. In the mouth, the digestion of protein does not occur in mouth since the saliva does not contain any proteolytic enzymes, so the digestion of protein starts only in stomach. In the stomach, pepsin is the only proteolytic enzyme in gastric juice. Renin is also present in gastric juice but it is absent in humans. In the small intestine, most of the proteins are digested in the duodenum and the jejunum by the proteolytic enzymes of the pancreatic juice and succus entericus. Proteolytic enzymes in the pancreatic juice. Pancreatic juice contains trypsin, chymotrypsin and carboxypeptidases. Trypsin and chymotrypsin are called endopeptidases as these two enzymes break interior bonds of the protein molecules. Proteolytic enzymes in succus entericus. Final digestion of proteins is by the proteolytic enzyme present in succus entericus. It contains dipeptidases, tripeptidases and aminopeptidases. Final products of protein digestion. Final products of protein digestion are the amino acids which are absorbed into the blood from the intestine. Absorption of proteins. Proteins are absorbed in the form of amino acids from small intestine. The levo amino acids are actively absorbed by the means of the sodium co-transport. The dextro amino acids are absorbed by the means of facilitated diffusion. Absorption of amino acid is faster in duodenum and jejunum and slower in ileum. We can observe the below diagram how the proteins are digested as the final product. Disorders of GIT. There are 6 types of disorders. First one, gastritis, gastroentropy, peptic ulcer, hepatitis, gallstones, Crohn's diseases or enteritis. First one, gastritis. The inflammation of gastric mucosa called gastritis. It may be acute or chronic. Acute gastritis are characterized by the inflammation of superficial layers of mucous membrane. Chronic gastritis involves inflammation of even deep layers of infiltration with more lymphocytes. Second one, gastroatrophy. It is the condition in which the muscles of the stomach shrink and become weak. Gastric glands are also shrink, resulting in deficiency of gastric juice. Third one, peptic ulcer. Peptic ulcer means the ulcer in the walls of the stomach or duodenum caused by the digestive action of gastric juice. Fourth one, hepatitis. It is characterized by swelling and inadequate functioning of the liver. Hepatitis may be acute or chronic. Fifth one, gallstone. Gallstone is a solid crystal deposit that is formed by the cholesterol, calcium ions and bile pigments in the gallbladder of bile duct. Next, sixth one, Crohn's disease or enteritis. Enteritis is an inflammatory bowel disease, IBD, characterized by the inflammation of small intestine. It affects the lower part of the small intestine, the ileum. The inflammation causes malabsorption and diarrhea. Next topic, energetics. Points to be covered in this topic: formation of and role of ATP, creatinine phosphate and basic metabolic rate. Formation and role of ATP. Introduction. Metabolism is an energy balancing act between the catabolic decomposition reaction and anabolic synthesis reaction. The molecule that participates most often in energy exchanges in the living cells is called adenosine triphosphate. It is also known as ATP, which couples energy releasing catabolic reactions to the energy requiring anabolic reactions. ATP, adenosine triphosphate, or ATP, is the energy currency of living systems. ATP transfers energy liberated in exogenic catabolic reactions to power cellular activities that require energy. Enderogenic reactions. ATP consists of 3 phosphate groups attached to the endosine, a unit of composed adenine, and the 5 carbon sugar ribose. When a water molecule is added to the ATP, the third phosphate group is symbolized by the P in the following discussion is removed and the overall reaction liberates energy. The enzyme that catalyzes the hydrolysis of ATP is called ATPase. The removal of third phosphate group produces the molecule called adenosine diphosphate. In the following reaction, ATP, adenosine triphosphate treated with water gives adenosine diphosphate plus phosphate group plus energy. The energy supplied by the catabolism of ATP into ADP is constantly being used by the cell. As the supply of ATP at any given time limited, a mechanism exists to replenish it. The enzyme ATP synthase catalyzes the addition of the phosphate group into the ADP. ADP plus phosphate group plus energy gives the ATP plus water. The energy needed to attach the phosphate group to ADP is supplied mainly by the catabolism of glucose in a process called cellular respiration. Cellular respiration has 2 phases: anaerobic respiration, aerobic respiration. Anaerobic respiration in a series of reactions that do not require oxygen. Glucose is partially broken down by the series of catabolic reactions into the pyruvic acid. Each glucose molecule that is converted into a pyruvic molecule yields 2 molecules of ATP. Aerobic phase. In the presence of oxygen, glucose is completely broken down into the carbon dioxide and water. These reactions generate heat and 36 or 38 ATP molecules. Mechanism of adenosine triphosphate formation. The high energy phosphate bond that attaches the third phosphate group contains the energy stored in this reaction. The addition of phosphate group into a molecule called phosphorylation increases its potential energy. Organisms use 3 mechanisms of phosphorylation to generate ATP, adenosine triphosphate. Substrate level phosphorylation generates ATP by transferring a high energy phosphate group from an intermediate phosphorylated metabolic compound, a substrate, directly into ADP. In human cell, this process occurs in cytosol. Second one, oxidative phosphorylation removes electrons from the organic compounds and passes them through the series of electron receptors called electron transport chain to molecules of oxygen. This process occurs in the inner mitochondrial membrane of cells. Third one, photophosphorylation. It occurs only in chlorophyll containing plant cells or in certain bacteria that contain or other light absorbing absorbing pigments. Role of ATP. Role of ATP in linking anabolic and catabolic reactions. When complex molecules and polymers are split apart, catabolism at left, some of the energy transferred from the ATP and the rest of the given off as heat. When simple molecules and monomers are combined to form complex molecules, anabolism at right, ATP provides energy for the synthesis and again some energy is off as heat. The below flowchart shows the simple molecules such as glucose, amino acid, glycerol and fatty acids, how it is distributed in catabolic reactions and anabolic reactions into complex molecules like glycogen, proteins and triglycerides, how the heat is released in from the left and the right sides are given. The cycle of ATP forms into ADP with third phosphate group also is given. Please observe this flowchart. Second one, active transport. ATP plays a critical role in the transport of macromolecules such as proteins and lipids into and out of the cell. The hydrolysis of ATP provides the required energy for active transport mechanism to carry such molecules across the concentration gradient. Third one, cell signaling. ATP has a key functions, both the intracellular and extracellular signaling. ATP is also involved in the signal transduction. Its phosphate groups are used to the kinases in the phosphate transfer reactions which activate the cascade of protein kinase reactions. Fourth one, structural maintenance. ATP plays a very important role in preserving the structure of the cell by helping the assembly of cytoskeletal elements. It also supplies energy to form the flagella and the chromosomes their appropriate functions. Muscle contraction. ATP is critical for the contraction of muscle. It binds to myosin to provide energy and facilitate its binding to actin to form a cross bridge. ADP and phosphate and then release ADP are then released. A new ATP molecule binds to the myosin. This breaks the cross bridge between the myosin and actin filaments, thereby releasing myosin for the next contractions. Sixth one, synthesis of DNA and RNA. During DNA synthesis, ribonucleotide reductase reduces the sugar residues ribonucleic acid diphosphates to form the deoxyribonucleoside diphosphate ADP. The adenosine form ATP is a building block of RNA and it is directly added to the RNA molecule during RNA synthesis by RNA polymers. The removal of pyrophosphate provides the energy required for this reaction. Next, creatinine phosphate. While the muscle fibers are relaxed, they produce more ATP than they need for the resting metabolism. The excess ATP is used to synthesize creatinine phosphate, an energy-rich molecule that is found only in muscle fibers. The enzyme creatinine kinase catalyzes the transfer of one of the high-energy phosphate groups from ATP to creatinine, forming creatinine phosphate and ADP. Creatinine is a small amino acid-like molecule that is synthesized in the liver, kidneys and pancreas and then transported into the muscle fibers. When the contraction begins in the ADP level, when the contraction begins and the ADP levels start rise, creatinine kinase catalyzes the transfer of one high energy phosphate group from creatinine phosphate back to the ADP. This direct phosphorylation reaction quickly regenerates new ADP molecules. The creatinine phosphate and ATP provide enough energy for muscle to contract maximally for about 15 seconds. This amount of energy is sufficient for the short of burst of activities. Once inside the cells, it is transformed into the phosphocreatinine by the enzyme complex creatinine kinase. which makes it able to donate its phosphate groups to convert adenosine diphosphate into the adenosine triphosphate. This process is an important component of all vertebrates' bioenergetic systems. Creatinine can be broken down into the creatinine, which is then excreted in the urine. Basal metabolic rate, the overall rate at which the metabolic reaction use energy termed as the metabolic rate. Some of the energy is used to produce ATP and some is released as heat because of many factors affect metabolic rate. It is measured under the standard conditions within the body in a resting and fasting condition called basal state. The measurement obtained under these conditions is the basal metabolic rate BMR. The most common way to determine BMR is by measuring amount of oxygen used per kilocalorie of food metabolized. When the body uses 1 liter of oxygen to oxidize the typical dietary mixture of triglycerides, carbohydrates and proteins, about 4.8 calories of energy is released. Factors affecting metabolic rate: exercise. During heavy exercise, the metabolic rate may increase as much as 15 times of basal rate. Hormones. Thyroid hormones are the main regulators of BMR. BMR increases as the blood levels of thyroid hormone rise. Thyroid hormone increase BMR in part by stimulating aerobic cellular respiration. As cells use more oxygen to produce ATP, more heat is given off and the body temperature rises. Other hormones have minor effects on BMR. Testosterone, insulin and human growth hormone can release the metabolic rate by 5 to 15%. Nervous system. The sympathetic division of autonomic nervous system is stimulated during exercise. In a stressful situation, its postganglionic neurons release norepinephrine and it also stimulates the release of hormones epinephrine and norepinephrine by the adrenal medulla. Both the epinephrine and norepinephrine increase the metabolic rate of body cells. Next, body temperature. The higher body temperature, the higher metabolic rate. Each 1 centigrade rise in core temperature increases the rate of biochemical reactions is about 10%. As a result of metabolic rate may be increased substantially during a fever. Ingestion of food raises metabolic rate 10 to 20% due to energy costs of digesting, absorbing and storing nutrients. This affects food-induced dermatogenesis is the greatest after eating high-protein meal and less after eating carbohydrates and lipids. Age. The metabolic rate of a child in relation to its size is about double that of elderly person due to high rates of reaction released to the growth. Other factors. The other factors that affect the metabolic rate are gender, lower in female, expect during pregnancy and lactation. Climate, lower in tropical region, sleep lower and malnutrition lower. For more updates, subscribe to our YouTube channel Pharma Pro EDU. Thanks for subscribing. Meet again.